Hey everyone, and welcome to our latest explainer. I am absolutely thrilled to have you here today, especially those of you in our clinical audience, because honestly, we are looking at a total paradigm shift in how we understand human biology. We're going to be talking about a landmark 2026 study published in the journal Nature. And this study, it completely reframes a molecule called spermine. Now, for a long time, we've known about spermine's role in cellular regeneration, right? But this new research reveals it as our body's ultimate natural defense against highly reactive toxic iron. We're going to unpack exactly how this works, why our natural defenses so often fall short, and how a targeted nutritional ecosystem might just be the key to restoring this vital biological shield.
Okay, let's dive into this. Here is our clinical roadmap for today. We'll start with the 2026 spermine breakthrough. Then look at the threat of reactive iron, explore the 3xBiotics fermented ecosystem, detail the mechanisms of iron control, discuss severe iron overload pathologies, and finally, touch on silent aging and iron accumulation.
Section 1, the 2026 spermine breakthrough. Let's set the stage with the science that sparked this whole conversation.
So, 2026, this was a massive wake-up call for the scientific community when it comes to cellular protection. This groundbreaking study in Nature identified spermine not just as a standard polyamine, but as a critical endogenous chelator of ferrous iron, and more specifically, as a natural inhibitor of a devastating process known as ferroptosis.
Now you might be wondering, what exactly is the enemy we're dealing with here? Well, it's ferroptosis. That's a type of programmed cell death driven by iron-dependent lipid peroxidation. And the primary culprit behind it? Fe2+, or labile toxic iron. This is a highly reactive fraction of iron that is essentially just roaming free, causing massive oxidative stress and literally tearing through cellular structures. What's really interesting about the 2026 study is that it showed how spermine naturally binds to this dangerous reactive iron fraction. It neutralizes it and prevents cellular destruction. It acts like a giant biological sponge for toxic iron.
Moving on to Section 2, the threat of reactive iron. Because if our bodies naturally produce spermine, why is this still such a huge problem?
Well, the problem is what we call a biological bottleneck. You see, spermine is present in all our cells, but the vast majority of it isn't just sitting there in some permanent reserve ready to go. It has to be continuously produced through the physiological conversion of another polyamine called spermine. So our protection doesn't just depend on the static amount of spermine we have right now, it relies entirely on our body's active, ongoing capacity to transform spermine into spermine. Without a constant supply of spermine and the specific metabolic cofactors needed to make that conversion happen, this cellular assembly line basically grinds to a halt and reactive iron runs rampant.
Which brings us perfectly to Section 3, the 3xBiotics fermented ecosystem. Let's look at a targeted solution.
So how do you actually build a biological defense system from the ground up? It takes a highly specific matrix, like the supernutrients 3xBiotics formula. This isn't just one magic ingredient. It's a matrix of 3 complementary fermented systems plus a really powerful polyphenol source. First up, we have Armida, which is polyfloral pollen fermented in SCOBY. This is your biological core, providing that absolutely crucial spermidine. Second, Apikefir SCOBY, that's sequentially fermented apolernal, delivering essential precursors like methionine, arginine, and ornithine to fuel the cycle. Third is Cholkefir, a fermented colostrum supplying lactoferrin and highly bioavailable peptides. And finally, Aronia melanocarpa, which is just a powerhouse of antioxidants. Every single element here contributes a specific vital molecule to the assembly line.
Let's look a little closer at that last component, Aronia melanocarpa. It doesn't form spermine directly, but it acts as the ultimate backup defense. While spermine is busy binding up that reactive iron, Aronia's rich profile, we're talking anthocyanins, proscyanidins, camphorol, chlorogenic acid, and quercetin, steps in to strictly limit lipid peroxidation. And remember, that is the core trigger of ferroptosis. It even activates the NRF2 cellular response. It's a perfectly engineered tag team of oxidative protection.
Section 4, mechanisms of iron control. How do all these moving parts actually work together?
Let's sequence the exact chronological journey so you can see how this builds step by step. Step 1. The spermidine supplied by the Amrita complex converts into spermine, fueled by those amino acid precursors. Step 2. This newly minted spermine encounters the dangerous labile Fe2+ and temporarily neutralizes its toxicity. Step 3 is super crucial. Spermine isn't a storage facility. Actually, scratch that. It's not a storage facility at all. It's a handoff point.
It safely hands the iron off to the body's natural transport proteins, like ferritin, transferrin, and importantly the lactoferrin provided by the cholecalciferol. And the result? Step 4. The iron is safely managed and the cell is completely protected against ferroptosis. 5. Severe iron overload pathologies. Let's look at the clinical reality where this mechanism is tested to its absolute limits.
In severe conditions like thalassemia, hemochromatosis, myelodysplastic syndromes, sickle cell and aplastic anemias, the body is completely overwhelmed by toxic Fe2+. Whether it's from repeated blood transfusions, chronic hemolysis, or hyperabsorption from the diet, the natural equilibrium of iron is just shattered. The sheer volume of reactive iron circulating in these patients actively damages the liver, the heart, and the endocrine system. In these severe hematological states, spermine's natural chelating mechanism isn't just a nice-to-have bonus, it is an absolutely vital physiological shield desperately trying to hold back a flood of oxidative destruction.
Section 6. Silent aging and iron accumulation. This is where the story shifts from rare diseases to a very common threat.
We really need to contrast acute iron overload, which is severe and generally pretty easy for a clinician to spot, with silent accumulation. For the vast majority of people, iron accumulation happens silently, progressively, and completely without symptoms over decades. Think of it like microscopic rust silently weakening the structural integrity of a building. As we age, our natural regulatory mechanisms decline, and this rust just slowly builds up in our tissues, acting as a slow-burning fire of oxidative stress.
So who is actually at high risk for this silent threat? Well, men over 40 to 50 and postmenopausal women are right at the top of the list, simply because they lack regular physiological pathways to eliminate iron. But we also see massive iron accumulation driving chronic inflammation in metabolic syndrome, insulin resistance, and MSLD, that's metabolic fatty liver disease. Even really popular high heme iron diets like heavy keto or carnivore diets can accelerate this whole process. When we lose the ability to naturally eliminate and safely store iron, it leads directly to tissue fibrosis, atherosclerosis, and accelerated aging.
And this reactive toxicity, it doesn't stop at the liver or the heart, it goes straight to the brain. Abnormal iron accumulation is strongly observed in the regions of the brain associated with cognitive decline in Alzheimer's disease, as well as in the substantia nigra in Parkinson's disease. This highly reactive iron heavily amplifies neuronal damage and age-related mitochondrial dysfunction. It just perfectly highlights exactly why a continuous, uninterrupted production of spermine to neutralize this toxic iron in the brain is absolutely critical for healthy longevity.
As we wrap up this explainer, I want to leave you with a final thought that really captures the essence of that 2026 Nature study. Spermidine is not just a molecule. It is a dynamic, active part of a physiological defense system that protects us from the heavy toll of time and disease. But it requires the right precursors, the right metabolic environment, and the right complementary antioxidants, like those we saw in the 3xBiotics ecosystem, to keep that defense system running. It truly makes you think, how well equipped is your body right now to handle the silent accumulation of iron? Thank you so much for joining me to explore this fascinating breakthrough. Keep questioning, keep learning, and I'll see you next time.